Hello everyone, we're back again. For now, we're going to go through the immunity system. As we said previously, that the immune system here is more likely as a first defense mechanism inside of your body. That means that any sort of microorganism or anything that gets or tries to get into your body, it would be classified as a foreign body, something strange, something the body is not used to. So what does your body do in order to prevent that sort of microorganism from getting inside and causing illness for you? The system here, or the body here, would actually have specific sort of cells that they will make the first line defense. Let's go through and discover what they are. The immune response here, the body's immune system, responding to non-self antigen by what? The lymphocyte and the phagocyte. Remember these two, so we will know them later on. It involves the production of antibodies and the killing of cells that have become infected by the pathogens. The phagocytes are produced and stored where exactly? In the bone marrow. I believe in grade 7 and 6 you came across that one of the skeleton system's functions is actually production of blood cells. Where exactly? In the bone marrow. Through life before being distributed around the body by blood. They remove the dead cells as well as invasive microorganisms. Then we have another sort of blood cell which is called neutrophils. It makes up 60% of the white blood cells. So the first thing that you know already, that the blood cells, we've got the red blood cells and we've got the white blood cells, and each white blood cells, since the white blood cells are the main cells or the main blood cells that they actually are carrying the process of the immune system or the immune response inside of your body, so it has like several cells classified beneath it. One of them that we just mentioned, the phagocytes and the lymphocytes and also the neutrophils. The neutrophils here happen to make up 60% of the white blood cells. They circulate the body and leave through what exactly? The capillary walls to enter the tissue during the infection. One of its properties, it's actually short-lived and often die after ingestion and destroying bacteria, forming pus. What about the macrophage? They are a mature form of monocyte that have left the blood and settled in organs. They live a bit longer and they're larger than neutrophils. Where can I find them? I can find them in the lungs, the liver, the spleen, and kidney, and also the lymph nodes, where they actually engulf the foreign particles and microorganisms. If you remember that we said once we have the foreign body or the foreign substance that has tried to invade the body, the macrophage's function is actually to engulf this foreign body to allow the lymphocyte afterwards to analyze and destroy the core of the microorganism or actually the foreign body. Also known as antigen presenting cell, which is shortcut for APCS, as they ingest antigens and display them on the surface, allowing the T lymphocyte to bind to the antigen and stimulate the immune response. Okay, the process of the engulfing, what do we call it? We call it phagocytosis. So during an attack by pathogens, cells produce chemicals called histamine, which attract neutrophils to the site of infection, which we call it by then chemotaxis. Okay. The number of the white blood cells increase at time of infection. If you have you suddenly got ill or you have an infection, what are the things more likely to happen to you? Sometimes you would just notice a bit of a lump behind your ears, or sometimes just below the lower jaw, you will slight feel an elevation and it's kind of hot. So this is like an early sign for you to know that there's something wrong inside of your body. There's actually infection. If you would wished to make sure, you would just go and run a blood test, and by then they will come to see the amount of the WBC that you have. When it's slightly above the normal, that means you have an infection inside of your body. The neutrophils during bacterial infection and whenever tissue becomes inflamed and die. Also, the lymphocytes in the blood increase in viral infections and tuberculosis. Most of the lymphocytes that circulate in the blood are T cells. The HIV invaders helper T cells and causes their destruction. So blood test for people who are positive for AIDS or HIV, they record the numbers of T cells specifically. Leukemia. What is leukemia? I'm pretty much sure that you've heard this term before by a lot of your maybe friends, relatives, or whatsoever. Leukemia is actually cancer like we call it blood cancer. But which sort of cancer? Cancer of immature white blood cells, disrupting the normal levels of RBC or platelets, causing anemia and risk of excessive bleeding. The mature WBC decreases so that people become more susceptible to infection. They are said to be immunosuppressed. That means they have low immunity. Here we've got another type of leukemia which is called myeloid leukemia, which is a cancer of the stem cell. The stem cells here, they are the originated cells that we have, and more likely nowadays that in America and Europe in general, they are working a lot of experiments on the stem cells to know that maybe they can cure cancer since they are the healthy originated cells that our body possess. That gives a rise to neutrophils, red blood cells, platelet and monocytes. The lymphoblastic leukemia, the cancerous cells, are those that give rise to the lymphocyte. And acute, that means it's very, very strong and it just comes once, like it's not repetitive. Develop very quickly and have severe effects and need to be treated immediately after diagnosis. Meanwhile, the chronic, that means it comes, for example, now and it shows up 5 years later, and then it's like it doesn't show up, and then after 10 years later it shows up again. That means it keeps coming and going. Chronic leukemia develops over many years. Changes in blood cell counts are monitored over time so that treatment is given. Okay. The lymphocyte. The lymphocyte is a white blood cell produced before birth where, like, this question is so repetitive, all the blood cells are originated in the bone marrow, because in the bone marrow, blood production is like occurs over there. Lymphocyte is a white blood cell produced before birth in the bone marrow. They are smaller than phagocyte and have a nucleus that fills most of the cell. We've got differentiations of lymphocyte. We've got B lymphocyte, we've got T lymphocyte. Okay? For the B lymphocyte, it's produced and developed in the bone marrow, concentrating where? In the lymph nodes and the spleen. When mature, each B cell produces one type of antibody. Part of the antibody molecules form a glycoprotein receptor that combines specifically with one type of antigen. Once combined, the B cell here goes under mitosis repeatedly to form a clone cell. That means just exactly the same. As only the B cells with antibodies complementary to antigen divide like this, it's known as colonal selection followed by colonal expansion, because it's more likely as presenting the exact sort of type from this cell. Some differentiate, that means they have specialization, into short-lived plasma cells, release antibodies into the blood, lymph linings of the lungs and the gut, which is referred here to the digestive tract or stomach specifically. And others become long-term memory cells, which is they have glycoprotein receptor on the cell membrane. Here, the plasma cell, there is an extensive network of rough endoplasmic reticulum in the cytoplasm for production of antibody molecules, which plasma cell secretes into blood or lymph by exocytosis. The mitochondria here, since it is a power or a source of energy, it provides the ATP, an energy for protein synthesis and the movement of secretory vessels. The T lymphocyte here, these cells are produced at the bone marrow as well, but where do they collect? In the thymus until maturity. The mature T cells have receptors that are complementary to the antigen of a pathogen. So it makes it much easier to recognize it, and in order to that, they destroy it. The T cells only respond when they encounter this antigen on the cell surface membrane of the host, like a macrophage. A particular T cell with a complementary receptor will bind to what? To the antigen, which is found on the antigen-presenting cell. So colonal selection and expansion also takes place and differentiate into the following. So the T lymphocyte differentiates into two things. Here we have the T helper cell and the T killer, the other name of it, cytotoxic. The T helper, what does it do? It secretes a hormone called cytokine, which stimulates the B and T killer cells to divide and macrophage to carry out the phagocytosis, which we said it is engulfing of the microorganism or the pathogen more vigorously. The T killer cell, or the cytotoxic T killer cell, it actually, what does it do? It destroys the cell to which they are bound, so they search the body for cells that have become invaded by pathogens and are displaying the pathogen antigen on their plasma membranes. When the T killer cells recognize the antigen, they attach themselves to it, or exactly to the surface of the infected cells, and secrete the toxic substance that actually kills the pathogen within them. Okay, this will be the last thing here. We've got several types of immunity. We've got the active immunity, which is a long-term immunity. Where does it, like, we gain it from an antigen once we have like a previous or a primary infection. So your body actually recognizes it. Whenever it happens again, they will have the exact antibodies to destroy it. The natural active immunity is obtained as a result of infection, where the body has manufactured its own antibodies, or the artificial ones that you actually took a vaccine towards it. The passive immunity is actually immediate but temporary immunity. You can take it like while you were still in your mummy's tummy. So practically here, it's immediate but temporary immunity gained without an immune response. So antibodies and antitoxins are injected, artificial, or passed from mother to child across the placenta or during breastfeeding. So no antigen is encountered, therefore no memory cells are produced. The vaccination is actually an antigenic material which could be alive or a dead or like a harmless sort of microorganism. You get injected to it, so it allows your immune system to actually produce antibodies. Therefore, whenever you get this microorganism to be invading your body, your immune system actually already recognizes it, so it sends right away the exact antibodies to fight it back. Less effective vaccines do not mimic an infection, as they are dead bacteria or virus that do not replicate inside of the body. They need a booster injections to stimulate secondary responses that give enhanced protection. But why sometimes those kind of people, they do get a vaccine, but somehow they have a poor response to it? Why? Because defective immune system does not produce necessary B and T cell clones, or those who actually suffer from malnutrition. They're not actually going through a proper diet. So by then, they don't have enough protein to make the antibodies. Live virus and herd immunity. We mentioned this before, that people may also be injected with a live virus and could potentially pass it out in their feces during the primary response. Evolved by mutation, the environment infecting others. This is why we vaccinate everyone at the same time, known as herd immunity. That means I have a group of people, I inject them with the same vaccine, all of them at once at the same time. So herd immunity interrupts transmission in a population so that those who are susceptible never encounter the infectious agents concerned. Like, for example, now we've got corona. So if they manage to come up with a vaccine or any sort of cure, they would be applying a herd sort of vaccination. Antigenic variation and antigenic concealment is not much to focus on. Smallpox eradicated because of what? Why smallpox is no longer present nowadays? Because the variola virus did not mutate and change its surface antigen. So the same vaccine could be used everywhere and it's cheap to produce. The vaccine was made from a harmless strain of the virus, the vaccina, and was effective because it was a live vaccine. Infected people were easy to identify, the vaccine was easy to administer, and the smallpox virus did not linger in the body after the infection. The virus here did not infect animals, which made it much easier to break the transmission cycle. But why measles is not eradicated? Because poor response to the vaccine shown by children who need several boosters to develop the immunity, and it can be difficult to give boosters follow-up cases of measles and trace contacts in large cities. And also the migrants and refugees can form reservoirs infection experiencing the epidemics with their community. So it will be actually much difficult for them to trace them back. And this is the end of this chapter.